Yeah, everybody, welcome this morning. I hope you had a good night's sleep and didn't drink too much last night. Really nice beer, actually, I think. Thank you very much for organising that, for the organisers. We're going to kick off today with a session on genetic and data resources and approaches for harnessing genetic diversity. And we've got 6 seminars this morning in this session. 2 of them will be online, so no questions. But we have 4 people live on stage telling us about genetics and data resources and approaches for harnessing diversity. So, Mats will introduce the first speaker.
Yes, and the first speaker, the keynote speaker is Nils Stein, research group leader at IPK Gattesleben and professor at Georg August Universität in Göttingen. Mr. DNA Sequence himself has been the main driver for pushing through all these small grain serial sequences, including barley. And now he's turning to the dark side. We will hear more about that. Please, Nils.
Thank you very much. Thank you very much to the organizers and also to this kind introduction. Agnes, I'm sorry, it's time now for taking over time. No, I try to be in time, of course. And thanks for showing up in that large number. That's the driest session in the early morning. Well, I was feeling obliged to agree quickly to give a speech in this session, and then the close of the meeting approached. I wondered, why did I agree? So I have to talk today now about the dark side of barley diversity, the gap between discovery and user-friendly access. And I added in brackets the sustainable use, because that's maybe the take-home message in the end. I want to encourage you to plan better your experiments, that we can sustainably use also the data you generate. So, since I was working since maybe 15 years now, or more even, in generating resources for the barley community, starting with ESTs, then with genome sequences, generating such data for community is a blessing, but it's also a curse. Blessing because you can use it for your research, a curse for the ones that have to provide the data publicly, easily accessible, and sustainably accessible. So we produced data over the times with the different versions of the barley reference genome, and as Martin outlined in the beginning of this symposium, we are sequencing just many more genomes. So it's more and more data that is accumulating, and this pyramid is just summarizing the status of the Pangenome project with draft sequences, skim sequences, but also complete genome sequences, large data sets that are not yet all in the public, but will become public in the next months as we are working on the second version of the barley pangenome. So, when we published a version of of a barley genome, often people approached me through email or on conferences, is the genome sequence data, is it available public? And I said immediately, instantly, yes, of course, because we really tried hard to make the data available. And I wondered why they didn't read the line at the end of the paper where data accessibility is all outlined. But the problem is that the people often have to rephrase their question, because they were not asking for the sequence, they were asking for the whole package. They asked for the full annotation with all details and best mapping of gene models among different versions and so on. We had also in previous discussions during the meeting requests, why isn't it possible? Well, and then my answer is a bit slower, because I have to admit that maybe things are not yet immediately user-friendly available, because this would require another funding of the dimension of a research project we just spent the money. So it's complicated. There are other questions I deal with, and so I mentioned already the question, why are the gene identifiers between versions not necessarily mapped to each other? Simple thing is that not every gene is present in every assembly. These are technical reasons. So I usually answer, yeah, please talk to Klaus Meyer and Manuel Spannager, who we collaborate with for the annotations, but it's more complex. Yeah, it's not that easy to do the mapping always precisely. And so it is a research question, basically. And then we heard the question also during the symposium, why is there no TARE? So for those of you who have never worked with Arabidopsis, TARE stands for the Arabidopsis information resource. And the reason there's no tear is if you go to the tear webpage, these are just screenshots of the different dimensions of that resource. You have the pull-down menus, which are too small for me to read even, but you have all the data related to Bali, to Arabidopsis there. And it is just a resource that was funded by the NSF for 15 years. Yeah, there was it's just enough funding to produce such a database. And then when NSF started to think about if they want to continue spending that money over ages, the community in Arabidopsis built a model of collecting subscription revenues. So they found a model to finance that resource also in long term. So what I want to do now in the next few minutes is to review with you a bit what is the status of data information for Bali in different contexts to genome resources in the past, in the present, and what we may need in the future. So start with the past. For those of the older semesters among us here, you know that in the early times, the main resource about Bali was the Bali Genetics newsletter. And you can go to Grain Genes and you will find all the volumes, and also among the younger ones. If you've never seen a volume of the Bali Genetic newsletter, I clearly recommend you to go there, because you will find all the information of the descriptions of the Bali genes, mutants, and so on and so forth. But you will also get a flavor of how science communication in a community like this happened decades ago. We will tomorrow or tonight in the business meeting also talk about the question whether there's a need to maintain this newsletter. Is it a format that still has a value? But Grain Genes was in the beginning also the central platform, not only for Bali, but for all the small grain cereals, mainly for information resource. Sorry, I took mainly screenshots for this. Don't try to read all the small font, but go to maybe the URLs, or I can send you also the presentation afterwards if you want to have the links to the data resources. Grain Genes was the main hub to access genetic map information, because long time we had no genomes, and it was the main site to go for genetic map information. Then as sequencing started first on transcriptome level, when cDNA libraries were systematically sequenced at the beginning of the 2000s, then project-specific databases showed up. Harvest, a project that was driven by Tim Close at UC Riverside, a very useful resource. At IPK, we had also an EST database. There were publications about these databases, and these databases faded away also again because the importance of EST databases is not longer there. There were other project-specific databases like the BALEX database, which we used to visualize the physical map information. The physical map was used to develop the first reference sequence of Bali, but this was of temporary use. The database is still accessible though. So with this short glimpse about more historical data repositories, what is out there now? And I will talk first about genomes, transcriptomes, proteomes, and later also we had already yesterday a session about germplasm. So again, Grain Genes, I think, is still the most complex or the starting point maybe to gather information about Bali genetics and genomics resources. And it's the team of Tanner Sen, Gary Lazo, Victoria Blake, her name was mentioned already yesterday, Eric Zhao, and Steve Michael. And yeah, you find a lot of information related to small grains, not only to Bali. And Grain Genes is the one place that is maybe as much as similar as possible for the small grains compared to TARE, because there is a long-term funding by the US Department of Agriculture. So it's a database for Triticale and Avena for molecular and phenotypic information, and it has a long-term perspective. How long, I don't know, and the question is whether also the community should make an effort and raise the voice also for the importance again, and maybe find additional parallel funding resources. If you go to Grain Genes, for genomes, you have these quick links. You can click on genomic browsers or genome browsers, and then a whole list of the different genome browsers for barley, wheat, and rye, and also oats is popping up, and you can go there. You have the JBrowse functionality there with the lowest level of the DNA sequence, and then you can load additional layers for annotation, so you can visualize then the genes along the chromosomes at different resolution. You can use also clicking on information there. There are cross-links to other databases. You can also download information from there. It's quite convenient. There is a seminar by Vic Carollo, which he may record again in full length, which will be available through the Bali Hub, James Hutton. Another similar resources at Ensemble Plants. Ensemble at EBI is developing these genome browser views on many genomes, and also Bali is there because that's one thing we immediately do when we have another assembly. We send these assemblies to Ensemble and to Grain Genes to have them there at the time of publication. So you have species-specific views there where you can then have the genome browser, but also Ensemble provides additional links for Synteny research and many other features. Go there and try. And then there are also, again, more project-related activities. We felt obliged to make data available also pre-publication through a BLAST server. So we have a Galaxy BLAST site at IPK where we provide also pre-publication access usually to BLAST against unpublished genomes of the Pangenome projects. And of course, what is important to mention is the resources that are available through the James Hutton Institute and in the future probably through the Bali Hub. The James Hutton Institute was very active in producing web resources to visualize and analyze, also, and also localized local applications. So they have developed SNP databases and document UK activities in genome sequencing. But more importantly, maybe more familiar to you are desktop applications like Germinate, Helium Tablet, Flapcheck, Curly, Whirly, and Strudel. These are all very nice visualization tools for different type of genomic information. Some of you may have used it already. One important database I want to mention is specifically from James Hutton is the EORNA. It was mentioned also during the conference already. It is a database to visualize transcript data from many hundreds, if not thousands, data sets from the public and own data at James Hutton, where you can immediately access also the differential splicing information. It's all based on the reference transcript data set that was developed there. So I think this is a data resource that will have a strong impact on the Bali community. A database that is maybe not as familiar to every one of you is the effort at UWA in Perth, led by Harvey Miller. It's on proteomics, and it's really worth a visit because they are trying to really provide for every protein in different species for which there is a subcellular localization available, either through experiments or through homology in other species. You can query for your gene model and get a subcellular prediction of the localization, and that can be quite informative for your experiments. I mentioned that some of the database resources are project-specific. This is one example. A paper on the Bali variation database. I tried to access that link. It's not possible anymore. This is what happens often to project-related databases, that when the funding expires, there is no maintenance. And I think this is what you really have to consider when you are planning to maybe invest time also in developing your own resources. What is important, there is a central database at EBI as well, the European Variation Archive. It's definitely important that when you generate such data, make sure you submit your variation data to this central repository, which is maintained long-term. Okay, for germplasm, we have talked about yesterday. You can go either to Eurisco to access the individual Bali datasets from European gene banks, or you can go to Genesis, which is the more international central hub to access genetic resources, datasets from different gene banks worldwide. You will see that there are hundreds of thousands of Bali accessions in the central repositories, and it's your way to, or your work to make your way through. And we heard about also this fantastic resource at NordGen, where all the genetic stocks are stored. So compared to TARE, Bali, the data resources are really pretty much distributed. There's not really one central hub, and one has to be aware. Again, to mention here Germinate, that's a desktop application for handling large data sets on genetic resources. I think that's a very important development at James Hutton, which is also used by several groups of the CG centers to manage their genomics data for genetic resource collections. And we have also developed such a data visualization hub at IPK for the Bridge project. So if you haven't visited the site, maybe you go there once and also provide us with feedback, because we are very much interested in how useful it is. The website provides direct access to the whole Bali collection, and the molecular data is available through a SNP browser. You have the PCA analysis, which is interactive. You can do lasso selections of the subpopulations and bring it into context to phenotypic data, which is shown in some of the screenshots. And you can also directly go from there to the ordering system of the IPK Gene Bank. And we have such a repository also in Japan, the DDB, the National Bioresources Project at Okayama University, led by Kasato. There's the Japanese Gene Bank, and they have also done systematic EST sequencing, full-length cDNA sequencing. They've done sequencing of a few Japanese cultivars, and they have also started to genotype their entire Bali collection. And finally, to mention is also the TriDC Toolbox. That's a website mostly known to US researchers because it is a mandatory data hub and interaction site for partners that were involved in the USDA or the Department of Agriculture funded TriDC coordinated agricultural projects. So there are basically mainly data analysis tools and so on for the project. So to summarize, this current state is databases and data resources for Bali are really distributed and not available through a single site. A lot of information is linked to grain genes. And often these databases were part of projects. They were a requirement of projects, and that has an impact on their long-term sustainability and access. So the long-term resources that are in place, and I mentioned this, are not necessarily Bali specific, so you have to make your way through to find the Bali information. So what do we think about the future, and how can we make an impact to the future in that topic by our own research? And one example is here. When I was preparing for another talk at a different conference, I came across a database that I wasn't aware of, and that's at the National Human Genome Research Institute and EBI. They have established a GBAS catalog for human genome-wide association studies. And in fact, when I talked to the editor of a journal, they told me this is mandatory now for papers in nature journals, that you have to submit your GWAS metadata and all the related data to that repository before you can publish. And I think this is exactly what is needed, because how many studies were there already in Bali on biotic, abiotic stresses in different material, and all that just is done for the drawer and the paper. Yeah, but how is the sustainable access to it? There are, if you go to Web of Science, 230 results for Bali and GWAS. So there are quite a few data sets already published. And I was then very pleased to see that Grain Genes, especially Victoria Blake, has made an effort to curate data sets from the literature. So they have, in May 2020, added to the genome browser of Bali curated GWAS tracks from quite a number of publications on different diseases. So they have 576 QTLs for net blotch. Then they have other diseases for many different diseases, but also other agronomic traits and also mold quality. That's really a very important activity. And I bring this up here because I want that you change your mind about your own research as well, that you design your experiments in that direction, that this data becomes more reusable. So when you go to Grain Genes now, you can use the JBrowse for Bali, and you can visualize the different QTLs in context of your genome sequence. And that can be very intuitive, as you can see here by a stack of QTLs at a chromosome for NetWatch. So, as I said, the Grain Genes people went to the literature and curated it out of the literature. But this is not the way it should be. Yeah, this is really what eats up the time. And if the papers were designed in a way in advance that the data could be automatically queried from a database with the respective tools, that's what we need in the future. And that leads me to the topic of FAIR data. I don't know if all of you have yet been familiarized with this term. I hope you are. FAIR stands for findable, accessed, interoperable, reproducible. And it's basically a concept that should allow us to reuse research data sets over long term. Yeah, they were generated maybe today, but maybe in 5 years there are new research results that make it important to reanalyze older data sets. And then if these data sets are not really findable, if they are not interoperable with the new systems, if the documentation doesn't allow you to say that the experiments were reproducible, it's just impossible to reuse the data. But all the developments in using artificial intelligence and data collections through browsers tells us there is a high value in the possibility of reusing and reanalyzing data with new development of tools and with new algorithms. So it's important to think about the research data management in your projects, and you will be asked by funding agencies to provide a data management plan for your project. And I think this is crucial. When I review projects and I don't see any reasonable documentation how people want to document their data and maintain the data, this is a clear argument for me to not give an excellent rating for such a proposal. So you have to think from the beginning, from the planning of the experiment, through the collection of the data, the processing of the material, the analysis, then the storage and maintenance of the data, the sharing and the reuse. You have to think about how to document your experiments, your data, your material. And there are already tools available that allow you. You can send your information and your metadata to individual repositories. This is just an example in context of sequencing data generation. But the very important thing is already much at the beginning. Think about your material you're working with. Yeah, you get an accession from your gene bank. Make sure you register the material you're working with to the biosamples database. You get a unique identifier, and then you can relate all the experiments to that identifier, and that allows also then interrelating and connecting experiments that were published by other people if they use the same material, for instance. So there are publications in this context. There is a FAIR cookbook. So how can you design your experiments, and how can you plan your projects to really generate FAIR data. And I don't go here through all these charts. It's basically a recommendation when to submit your data to what repository and why this is an advantage to link other subsequent information and metadata. One example that came to my mind here during the meeting, we have published the bridge data a while ago. There's the PRIMA project led by Luigi Cadavelli and Ernesto, who take advantage of part of the bridge collection, as Ernesto had outlined yesterday. And there is this H2020 project agent I'm coordinating, where we are taking advantage of genetic resources from the same pool, and for which there is already a lot of data generated. And even the PRIMA and the Asian project have an overlap in their interest in generating abiotic stress evaluation on genetic resources. Because all the material is genotyped, ideally you can do then also integrated analysis across projects. So it's so important that these projects design their data generation and data management in a fair way, that we can have a chance to really take advantage and generate added value beyond borders of projects. And one final thing I want to bring up in this context, many of you have been really made alert about it. It's important, and that's why I emphasize also the importance to really, at the very beginning of your project, you make yourself aware of the process of data generation and make yourself aware, is the material legal you're working with? Especially if you're generating sequence data information, so-called digital sequence information, you may end up in a deadlock because the material was not legal under the Nagoya terms. And then it will be hard for you to publish your data. Yeah? And it's good if you're not realizing this after 3 years and your PhD student wants to graduate. So really make yourself aware, what's the status of the material you're working with. With this, I'd like to summarize. I looked into the faces. I saw that not too many fell asleep, so the topic seemed to be of interest. Data resources in Bali are available through distributed repositories with different lifespan. That's just a fact at the moment. And data curation, which is behind of really maintaining these resources for long term, is an underestimated essential need, and usually kills projects also because there is not enough manpower to do it. Long-term access to high-quality data is only possible if a community like the Bali community identifies the need to establish and follow research data management best practice and fair standards. And we have to maybe think also about agreeing how to enforce this. And there are ways. We could enforce it through peer review of proposals or papers. And this may be a pain in the ass for everyone who submits the paper or the proposal, but for the community, it in the end will provide a better resource of data. So plan your genomics, transcriptomics, QTL, GWAS study as it is a resources project. Don't put the design of your research data management plan to the last 5 minutes before submission of the proposal. This is the wrong approach. Yeah, you have to establish this as a routine in your institution. And different institutions provide different opportunities for this, I have to admit. And the question to the Bali community is whether we should also raise our voice to develop GrainGenes into something more of a TARE-like information resource. We could call it the TRIDC All Information Resource to have also a Bali TARE. The question is just if and how. And with this, I would like to acknowledge a few names. I have taken advantage of information on some of the slides from Vic Carollo and also from Uwe Scholz and Matthias Lange, but I want to thank the groups of Uwe Scholz and all his members at different research administration level, and also Martin Mascher, because they provide me at IPK with really an almost already ideal environment to make me aware about these problems, but also to help me in my research projects with all these aspects. And I'd like to thank the organizers for giving me the opportunity to talk about this topic. Thank you very much.
Niels, from your snapshot of what resources are available today, it sounds like we need an engine to find the correct engine as well, because there are a lot of students who so on who don't know how even to navigate through GrainGenes and find what it is exactly they need to use.
Yeah, well, I think it's rather straightforward to go through GrainGenes, but sometimes it helps also to talk to people, and that's been quick. But to some extent, you're right.
I mean, it was a great effort and a lot of work to connect all people to do the sequencing. But would it be even worse to connect people to organize the databases and create funding for that?
Yeah, I would agree. But of course, assembling people around a genome project for a while was a strong incentive, because there was maybe a paper in Nature or Science at the horizon, and people were stimulated. And for a database or community project to organize the sustainability or accessibility of data and resources is not necessarily recognized as a scientific activity. And it is the main thing is to have the necessary funding in place and convince the funding agency this is not just wasted money.
Yeah, it's more like building a highway.
Yes, exactly.
Maria?
Yeah, Niels, I actually had a very similar question. I was wondering if you could elaborate a little bit on what you think are the chances of a Bali TARE and what it takes, and we know what it's funding. And I guess you've already pushed a little bit or thought about it, how to put it in place, or you haven't, I don't know. Maybe you can elaborate a little bit on how you see the chances. And
no, it is something I have not developed further and hidden my concept behind.
okay.
Sorry for that. But I think important is firsthand that a community like this discusses this and evaluates the possibilities, because the community is organized in different entities. We have universities, we have non-university research institutes, we have companies. I think in TARE, they founded a company in the beginning to establish also a model for the financing, and I think this is maybe the Bali community is smaller than this community, which can make it more difficult. However, the applied importance of the crop may facilitate also the fundraising for money from industry. I think we need a discussion and try to find a consent. Maybe the community can work towards also a position paper on that respect, that we express our opinion and that we can forward this message then also to politicians and funding agencies to tell them, look, we would like to do the next step, but we can't do it without the necessary infrastructure support underlying it.
Thanks, Niels. Very interesting talk. Do you think this could be the topic of one of the expert working groups that we discussed on the first day, and then bring it forward, once it's discussed within the group, bring it forward to the community?
Absolutely. I would love to see that there are interested people coming together and forming such a task force to bring forward that idea. And yeah, would be great. And if you need names to connect with, I could also help. We could help, and Grain Genes would definitely also be interested to be part of it.
Put somebody in the CT as well.
Yeah, sorry, I missed the question there. So that will be the last.
Niels brought up a couple of good things. So I was just going to say on Nagoya, just to clarify that for the audience, of course, there's the International Treaty on Plant Genetic Resources for Food and Agriculture that, of course, in this case, when applied to Hordeum, covers things. So Nagoya, in this specific regard, luckily is, I mean, it's something you need to worry about, though, because suddenly projects get bigger, right? And there is the current discourse on how sequencing data applies to that as well, which could change.
You're right about Hordeum and the treaty and Nagoya, but the truth is that gene banks in the end give away material on the basis of the SMTA. Yeah, so it's not as simple as you say.
No, I agree. But I'm just for the audience.
Better to be aware of the
Yeah.
of all the difficulties attached to Nagoya.
Certainly for gene banks, it's an even bigger deal because of distribution, which is a major role that you have. I want to just touch on briefly, changing data over time is, of course, the biggest problem we have. And how many instances we've attempted to unify only to be undermined by a changing landscape of technology and strategy for that. So, and of course, even as we move into pangenome, how we're going to mitigate that and what would be the actual implementation of that from a bioinformatic perspective and how scientists access that is something we have to prepare for in terms of, that's a comment.
Yeah,
I guess.
no, I think that's very important. And if there is an expert group formed in the Bali community, I think it would be very important to reach out first to other communities who went through that process already and really orientate ourselves. What are the established approaches?
Yeah, and last comment briefly, just to bring this all together. Grain Genes is going to probably be pretty stable, and the way to maintain that stability is user use, right? And I think, in fact, that's probably in the community, aside also from IPK, in terms of having enough people working in it, would be one of the best things to develop. So probably thinking about how we would reimagine that would be a good strategy in the long run. And I mean, in contrast to Arabidopsis, we have, it's a commodity, right? So we actually have companies which Arabidopsis doesn't have, other than from a basic fundamental perspective that there are companies investing in Arabidopsis as a system for testing hypotheses and development. But so we have that.
There's maybe even also funding frameworks. There's the Foundation for Funding Agricultural Research in the US, who is providing matching fundings to existing research projects. Also on data management, they are very much interested, the Gates Foundation as well. So I think we have to just involve also Grain Genes, and I hope that was a message I wanted to give. We should maybe be aware better of Grain Genes and develop it further as a community.
Thank you for the introduction, and I would also like to thank organizer for providing opportunity to present our work on genetic architecture of grain site in Bali. So it's the main objective of cereal research is to improve the grain yield in terms of optimal and suboptimal conditions. And in our group, we work on both source improvement and also sink improvement to achieve this goal. And in terms of source improvement, we we mainly focus on photosynthetic efficiency and also the macro and microscopic traits associated with it. And when you talk about zinc improvement, one of the major trait is a grain number. And in barley, there are almost at least 5 loci that has been characterized that controls the lateral spikelet fertility using either mutant population or also natural populations. And the other aspect is grain weight, and which is mainly controlled by the zinc capacity or grain filling rate. And today I would like to talk about the phenotypic diversity and also the genomic loci associated with those traits in barley. For this, we use a multi-parent populations, and it was developed from the genetic cross between 23 diverse barley inbreds, and they are crossed in a double round robin scheme. So it means that one inbred is crossed with 4 other inbreds, and a random set of F2 lines were selved to produce 45 recombinant inbred line subpopulations, and each subpopulation has from 35 to 146 individuals. And so therefore around 4,000 reels were genotyped using 50K SNP, and the grain size parameters were evaluated from the field trials in 4 different environments from 2017 until 2020. So these are the grain phenotype of the parental lines for all 45 subpopulations, and you can see the variations for grain length here in this picture. And there are, for example, genotypes like ANCAP2 or 12830 Lakan, which has particularly long grains, and there are genotypes such as Namhebori K10693, which has slightly shorter grains, and other genotypes has in between phenotypes. So as a whole, you can see a huge variation for the grain length in this population. So here is just a numerical representation of the phenotypic variation for the grain size parameters for all those 4,000 recombinant inbred lines. And so when we looked at the range of the adjusted interim means for our real population, we found out that the range was slightly wider than reported in the previous linkage mapping studies. And also we found out that the grain size parameters are highly correlated, positively correlated with each other. And also, especially 1,000 grain weight and grain width was positively correlated with the grain yield per plot. And if you look at the table on the right, you can see the variance component for several factors, and we found out that the variance component due to genotype was almost 2 to 10-fold higher for genotypic component compared to environment. So this means that the variation for the grain size we observed is mainly due to the genotypic effect than the environmental effect. And this is also reflected in terms of broad sense heritability, which is 85% or higher for all grain size characters. In the next step, we performed a multi-parent population QTL analysis using a R package called MPPR, and we use parental model because it utilizes the information of the connections between the parents for each cross, and also it assumes that the effect of parent is constant across each cross. And with this, we were able to find QTLs for all 4 grain size traits distributed across 7 chromosomes of barley. And to do MPP analysis, we use a consensus map, and there might be some imprecision in the consensus map. Therefore, we also went back to single population analysis, especially to define the confidence interval of the QTLs. And as expected, we found subpopulation specific QTLs that clustered at the same locations of the MPP QTLs. And by this, we were able to find 62 consensus QTLs, which was the shortest possible confidence interval across the overlapping subpopulation specific QTLs. And we found out that at least 13 QTLs were not reported in the previous linkage mapping and also association mapping studies as related to the grain size characters described here. And there were also some loci that were co-located for all grain size characters. We also found some proof of concept loci because the population involved the cross between 2-row and 6-row barley, and this particular phenotype might have a confounding effect on the grain size. And we saw a strong signal at VRS1 locus on chromosome 2, and similarly, we also saw a strong signal at NOD locus, which was also described in the previous presentations. So we also have 7 subpopulations where there was a cross between a NACT barley and Hall barley, and when we checked because we also have the genome-wide variant calling data for all 23 parental inbreds obtained from the whole genome sequencing of these lines, and when we checked the structural variant data, we found this 16 kb deletion on the chromosome 7 in these NACT barley cultivars, which is also the characteristics of the all NACT barley accessions. So this tells you that the QTLs we detected are more or less accurate. We also do a QTL validation. For this, we chose one of the measured QTL on chromosome 1. And what we did was we selected one subpopulation as VDR33. It was developed by a cross between a long grain cultivar Lakhan with a short grain cultivar Georgie. And we picked 2 recombinant midlines that has a contrasting allele at the QTL, but they were almost isogenic in the other parts of the region, at least for the other QTLs. And then we phenotyped, first we genotyped the anthropogenies on the left and right border of the confidence intervals, and these are just the preliminary results. And if you see the non-recombinants, the plants that has Lakhan allele on both borders has longer grains compared to the plants that has Georgie allele. So this somehow confirms the accuracy of the QTLs, and so we can say that these are relevant QTLs which we can use in further studies to identify the underlying genes that regulates such traits in barley. In the next few slides, I want to talk about the QTL effect. So here, this histogram shows the distribution of the percent of explained variance by the QTLs, and the dashed blue lines, which is the first quartile of the distribution, it separates QTL into roughly into 2 broad categories. So the first category where the explained variance is less than 10%, it means that almost 75% of the QTL explain 10% or more variance. Then we also did genomic prediction using genome-wide markers, and we found out that the prediction accuracy of genomic prediction model were almost 20 to 30% higher than the analogous figures from the full QTL model. So we were rather disappointed because even with such a complex design and 4,000 individuals, there are a lot of minor effect loci which cannot pass the significant threshold. So it says that to capture those minor effect loci, you have to even use more complex design. So this is the distribution of the allele effect across the QTL. So it's allele effect means if you have a 1 QTL, you will have 23 effects because we have 23 parental lines. So these are the distribution of those allele effects. So the yellow one is the negative allele effect, and the blue one is the positive allele effect. So the take-home message from this figure is that if you see that most of the allele effect is concentrated towards 0. So it means that the phenotypic variation we see is mainly controlled by the large number of minor and moderate allele and very few number of large effect alleles. And this is the simplified version of the previous graph. Here we divide the effect into each single parents, and these are the cumulative effects, either positive or negative, and the number above the bars is the number of the loci. There are a lot of information in this graph, but what I want to point out is that we have 12 land races and 11 cultivars in our among those 23 lines. And when we compare the average positive cumulative effect based on the germplasm type, it was only slightly higher for the cultivars. And it means that and also, if you look at some of the land races which I highlighted here, they made a significant contributions in terms of the number of the positive alleles and also the cumulative effect. So, which also highlights the importance of land races for agronomic traits. And then this graph is the oversimplified version of those all informations. And here it represents the allelic series for each QTL across for each QTL for grain length here. And if you look at the QTL on chromosome 1. So you have the blue sets and the green sets. So the intensity of blue and green sets indicates the magnitude of positive and negative effect, positive and negative effect. And if you see, let's only take first for the QTL on the first QTL on the chromosome 1, you can see there are different sets of blue. It means that, and so what does this means is that the effect of each parent at this QTL is different. So you can see that there's not only one type of effect, but the magnitude of effect is varying. And this is also true for all other QTLs across the 7 chromosomes. So which means that we see a clear natural allelic series for all the QTLs. So the possible explanation means that there might be different, not one single difference in the alleles, but there might be several alleles for the for underlying gene for these QTLs. But if you have to do fine mapping for every single subpopulations to prove this hypothesis, or there might be linked genes that control the traits which we were not able to uncouple using the given set of marker density. So with this, I would like to conclude my talk with few take-home messages. The first message is that, as I mentioned, that at least 20% of the QTLs we found were not described in the previous literature, which means that the grain size trait is even complex than what we thought. And we also found that the phenotypic variation we saw in this population for grain size is mainly controlled by the minor and moderate effect alleles. And we also found that the land races cannot is not only, we can use land races not only to land races not only to look for the stress related genes, but also to look for agronomically relevant traits. And this is the beginning of now further work, where we want to look for the underlying genes, and we use approaches such as fine mapping to delimit the regions, and also utilize transgenic approach to validate the candidate genes. At the end, I would also like to acknowledge all the group members who has contributed to this work, especially our group leader Benjamin Stich, and there will be also one poster from our colleague Francis Cosenza. I think it's a poster number 64, where he will also demonstrate how we can utilize this population to understand the genetic regulation of other complex traits. And so we have several passport data for those for this population. So there are genotyping data for all 4,000 lines. The metabolomics data for the parental lines, and also the transcriptomic data for the parental lines, and the genome-wide variant calling data for all those parental lines, so that we welcome the barley community to utilize these resources if you find some relevant traits for yourself. This is all from my side. Thank you for your attention, and I am happy to answer your questions.
Aziz, congratulations, very nice work. My question is, did you try to understand which is the average length of a QTL based on base pair, based on physical distance? Because that could be probably helpful to understand whether your multiple alleles that you're finding for each QTL are actually coming from different alleles on one single gene or by multiple genes.
We have slides for that. So we also have the the confidence interval for all those QTL interval you can see here. So some of them are really narrow, and some of them are very large. So like they are extending along the centromere. So there we don't know, but there are the intervals which are narrow, and we also have some candidate genes for that, which we are already started working using the transgenic approach on those candidate genes.
Thanks a lot. Great talk and great work. I'm just wondering the difference between the 40% and the 70%. How much of that you think got to do with maybe epistatic interaction, and have you looked into this possibility?
So there might be also, yes, we also found some epistatic QTLs, some between the main effect QTLs and also with the minor effect loci. So it could be possible that those differences are also coming from epistatic interaction. So these are the preliminary results where we find some epistatic QTLs, but we have not still predicted how much variance is explained by this epistatic QTLs. So we have not modeled it yet. So it takes a lot of time because of the number of individuals. So this has to yet to be done.
Yeah, I wasn't asked to ask this question. It seems like you have a slide for every question, so that's that's great. Thanks.
Ernesto.
Thanks, Aziz. Nice talk. I was wondering whether you're going to continue, or your group is going to continue to trying to develop super grain barley, either by QTL stacking or by genomic prediction. It would be an interesting resource. Maybe not agronomically, but at least for research, for sure.
So we are currently doing field trials with all those recommended inbreed lines to check if you can really improve yield, because there might always be, like, you have bigger grain, but then when you talk about yield, then there's no difference. So we are trying to figure out to find the lines where you can find this break between the yield and this and the seed size QTLs. But as you said, we will also try to stack the favorable alleles in one single gene, one single plant, and try to. This is a nice. I mean, we have not thought about it, but this is something what we to look to do in future.
Last one, Gustavo.
Your last answer saved me one question, so thank you for doing this in the field now.
Yeah.
And for the average grain weight, you have the average. But for the data you showed us for the grain dimensions, you were with the average, but you have the actual distribution for every single data. Have you considered analyzing the genetic basis or the relevance of the spread of that variation in grain sizes for each particular case?
You mean like grain size for each sample, you mean, right?
You show us QTL for grain length, for
Grain
instance.
length, yes.
That grain length is the average length of all the grains in that particular case.
Yes.
But you have actually the data for every single grain. And then you have the dispersion or the diversity on grain sizes for each particular case. And with that diversity, you may consider whether the QTLs are affecting the potential grain size, if you consider, for instance, the top 10% of the sizes, or the actual realized grain size. And those things matter a lot for when you go to the field.
So this is very nice comment. I would say that it's we have not thought about in this direction to use the variance component as the trait for the QTLs, but this is a nice complement, and we can try that, of course. Yes.
Hello, everyone. First of all, I would like to thank the organizers for inviting me to present our work. My name is Utman Bouhlel, and I'm going to present our work entitled CGEAR Barley Breeding Toolbox, a diversity panel to facilitate breeding and genomic research in the developing world. The current challenges for barley production are mainly climate change and the rapid increase in the world population that overload the need for food and feed production, especially in the developing world. The efficient use of the new genotyping technologies and the genomic research applied to breeding have the potential to help attaining the production needed to feed the increasing population while becoming more resilient to climate instability and extreme climatic events. However, despite the reduced cost of these new tools, many breeding programs, mostly in developing countries, still cannot afford them, and as a result, a technological gap between the developing and developed world has arisen. Representative and diverse collections of genotypes are a primary resource to dissect the genetic control of traits of interest and meet the breeding goals through the identification of beneficial alleles. Several highly diverse barley populations have been assembled in the last 30 years, but only a few of them have been made available to the international community. Most of these populations consisted in core collections of major gene banks, international barley collections, and collections of modern varieties and elite germplasm. However, many of these populations are not representative of the barley varieties adapted to the developing world or are too large to be grown with limited resources. In addition, only a limited number of them take advantage of the new genotyping technologies that allow the use of a large number of representative and stable markers. For it, and building on the extensive expertise of ICARDA on material exchange, we aim to take advantage and make available to the international community an association mapping panel to serve as a barley breeding toolbox, especially for the developing world. The selected panel needs to be representative of the germplasm grown in and adapted to the developing world, to cover a wide range of genetic variability, and to be of public domain or international public goods to facilitate germplasm exchange and unrestricted use. To achieve our goals, we assembled and genotyped a global barley panel of 530 genotypes, mostly spring type, including elite lines, cultivars, and landraces from different countries. The panel consists in elite lines from the CGR Global Barley Breeding Program, cultivar from Canada, USA, Australia, Africa, Europe, and others, and landraces. The Global Barley Panel represents a wide range of barley end uses, root type, growth habit, and geographical and environmental distribution. The Global Barley Panel was genotyped using the Barley Amphinium iSelect 50K chip. And in order to estimate the genetic diversity and differentiation within the Global Barley Panel, we conducted a principal component analysis, neighbor joining clustering, and analysis of molecular variance. Also, we used the SPARE non-negative matrix factorization algorithm implemented in the LEA-R package to calculate the individual admixture coefficient. Also, we selected 4 SNP markers described in the literature to be associated with major flowering time genes in barley, mainly VRNH1, VRNH3, PPDH1, and HVCN. These 4 SNP markers were selected to assess the extent of genetic diversity existing within the 530 barley genotypes based on flowering genes. AMOVA tests were used to calculate the variation explained by each of the SNP markers and their 16 allelic combinations. Also, to select a core subset of 250 entries that represent the diversity of the population and at the same time provide an optimized set of entries for association mapping studies, we used the mean of transformed kinship implemented in the genetic subset R package. The MTK measured the kinship of the genotypes in a subset and identify the most similar sets of genotypes. Out of 43,461 scorable SNP markers, 92% were segregating in the global barley panel and 89% of them were retained after quality filtration using the parameters of minor allele frequency higher than 5% and missing data less than 10%. Also, linkage disequilibrium showed a fast rate of decline and the decay distance was approximately 0.3 megabase per. The results of principal component analysis and neighbor joining clustering showed that root type and geographical origin are main determinants of genetic diversity in our population. The first axis of the principal component analysis, explaining 8.6% of the genotypic variance, separated the genotypes according to their root type, being the 2 row genotypes located towards the negative side of the axis and the 6 row one in the positive side. Also, the neighbor joining analysis allowed identifying 8 main branches or subgroups that were also clustered together in the PCA. While most of these clusters are characterized by specific row type and geographical origins, the Icarda germplasm was present and in all identified subgroups. The genetic variation between and within groups was further explored through the analysis of molecular variants. The AMOVA revealed that the row type grouping accounted for more than 10.6% of the whole genetic variation, with 6 row genotypes showing slightly higher genetic variation as compared to 2 row genotypes, as reflected by the higher mean square. Also, the 6 row genotypes contained a higher number of polymorphic loci, while the grouping based on geographical origins accounted for 12% of the genetic variation. And among origins, the Icarda material was the most diverse since it contributed to a higher share of genetic variation as per the mean square and contained the highest number of polymorphic loci. We evaluated the genetic diversity based on 4 SNPs previously identified as linked to major flowering genes and their generated allelic combinations. In total, we identified 16 allelic combinations. The SNP linked to each VCAN explained the highest genetic variation among populations, similarly to all 16 allelic combinations together. Also, early flowering allelic combinations and mostly AC15, AC12, and AC16 were the most frequent and characterized mostly 6-rowed genotypes from Icarda, India, and Landrace's origins. Late flowering allelic combinations were frequent mostly in the genotypes from Europe, USA, to a lesser extent of Canada. However, the Icarda material contained all 16 allelic combinations, indicating high allelic variability that could provide adaptation to different environment and farming practices. We used the phylogenetic tree and the PCA to see the assignment of different allelic combinations to the different identified clusters. The results show that flowering loci are associated with the genetic structure of the population. The 2-row genotypes comprises entry carrying late or intermediate flowering allelic combinations, mostly from USA, Canada, and Europe origins belonging to the 2nd, 3rd, and 4th group respectively. On the other hand, genotypes from Icarda and Australia that are included in the cluster 1 carry early to intermediate flowering allelic combinations, while the 6-row genotypes are mostly grouped in the clusters 5, 6, and 8 that are mostly composed of entries from Icarda, India, and other origins are carrying early flowering allelic combinations. We can conclude that the material used in the analysis are carrying specific allelic combinations that are linked to their geographical origins. It is generally agreed that the principal sources of structure in diverse collections of barley germplasm are spike, row type, growth habit, and geographic origins. The results obtained through population structure, principal components analysis, and neighbor joining clustering analysis confirmed this separation and indicated 8 major sources of diversity. The subpopulations located on the negative side of the first principal component consisted of 3 subpopulations that split according to their origin. These 3 subpopulations are 2-rowed and comprising genotypes from USA, mainly the subpopulation 1. The subpopulation 2 comprised European genotypes, and the 3rd subpopulation included genotypes from Icarda and Australia. While the subpopulations located on the positive side of the first principal component consisted of 4 6-rowed subpopulations and 1 2-rowed subpopulation. The 2-rowed subpopulation, which is mainly the subpopulation 7, that is colored in yellow, consisted mainly of Icarda 2-row genotypes that are derived from the cultivar Cannella. And the subpopulation 4, that comprised 6-row genotypes derived from the cultivar Petunia. These 2 subpopulations are both issued from the CGIAR barley breeding program for Latin America. The 6-rowed fair barley genotypes from USA and Canada were mostly grouped in the subpopulation 5, while the 6-row genotypes from India were included in the subpopulation 6. The subpopulation 8 instead consisted in Icarda elite lines, mostly derived from the cross with the cultivar Rehan 03, which is a mega variety from the Syria programs released in several North African, West Asian countries and characterized by its drought tolerance. In spite of these specific sources, lines from Icarda program were present in all 8 subpopulations, showing the high level of the global genetic diversity in our program. So in summary, the CGIAR lines show the wide diversity and representativity of the whole population. This group of lines showed the largest number of polymorphic loci and together with the landraces covered most of the diversity in our panel. Moreover, the Icarda lines are present in all the subpopulations and contained all 16 allelic combinations of phenology genes. For it and for their representativity of the adapted material to the developing world, they were selected as the base of the CGIAR barley breeding toolbox. And 312 entries of public domains that are mostly from the CGIAR origin and a few cultivated landraces were selected to assemble a collection of diverse barley germplasm representative of the germplasm grown worldwide and with a particular emphasis on the developing world. We used the mean of transformed kinship method implemented in genetic subsetter to dissect a final set of 250 entries. This method measured the kinship of genotypes in a subset and identified the most dissimilar set of genotypes. The CGIAR barley breeding toolbox showed a good coverage of most of the PCA spectra, except for the most negative values in the first principal component, where most of European, Canadian, and US varieties are located. The CGIAR barley breeding toolbox subset continue to preserve a good balance between 2-row and 6-row genotypes, and entries from all subpopulations identified through population structure are included in the subset. The proportions are similar in the subset as compared to the global barley panel, except for subpopulation 1, 3, and subpopulation 6, in which the subset shows a smaller proportion as compared with the global barley panel. In the two graphs at the right of the slide, the filled bar represent the global barley panel and the dashed bars represent the CGIAR barley breeding toolbox. The three subpopulations 1, 3, and 6 are characterized by harboring entries from USA, Canada, and Europe, the three origins identified as some of the least related to Icarda material. Also, all 16 allelic combinations identified as the most important phenological loci are well represented in the CGIAR barley breeding toolbox. So to conclude, the CGIAR barley breeding toolbox represents the genetic diversity of the global barley panel, and it is a solid and diverse representation of the barley grown in the developing countries, but also significant to the developed world. The CGIAR barley breeding toolbox will be made available together with the phenotypic and genotypic data as an international public good under a standard material transfer agreement that allowed its free use for research, education, and breeding in order to underpin the genetic mechanisms of traits of interest for barley cultivation. This is a real picture of the CGIAR barley breeding toolbox. It has been multiplied from single spike in 2022 and will be made available for request from 2023. For request, you can contact Miguel Sanchez Garcia and Andrea Visioni. I left their emails in the screen. And also you are kindly invited to visit 200 lines of the CGIAR barley breeding toolbox that are planted during the field day visit. Thank you all for your attention. Please do not hesitate to send me emails if you have any questions. I would be happy to answer your emails. I would like to take this opportunity to thank my supervisors, Dr. Miguel Sanchez Garcia and Dr. Andrea Visioni, all collaborators and our lab members, and I wanted to acknowledge all sponsors that contributed to the funding to this research.
In developing the two-row type of barleys, did you distinguish between those that had a deficiency and those that were normal two-row?
So the ones that come from the program, I would say they are really two rows because they are already selected to be elite. I mean, this comes from the international nurseries. They are selected for high yield in all the countries, so they are real two rows.
When you are doing the genetic diversity studies, the number of assessments from different groups or different countries was not very good. I mean, you had a lot of lines from ICARDA and very little number from Europe, from India, and then you compare and you make the conclusions that everything is good in ICARDA material and not in other materials. I think it should have been better to make conclusions when you have the same number of assessments from each countries or each groups.
Yeah, I'm not sure if I follow the question. We definitely didn't want to say that it's not good, the other groups. We wanted to say that thankfully, to all the collaboration the ICARDA program had in the last 30 years, was a lot of intercrossing between all these programs. A bit less, obviously, with the European and the US programs, but still there. You can see it in all the admixture. So the program took advantage of all these lines from India, all these lines from the US and Canada, all the lines from Europe, and it brought it back to the adapted, took it back the background that was useful for us. But it's really definitely not that they're worse or anything. It's just that also when you breed for a widely diverse environment target, obviously you have more diversity. It's just a matter of you are breeding for more different environments.
It's also not so easy to get the same number of accession from all these countries. You know that there are limitations for germ plus interchange. Thank you.
It was supposed to be a poster presentation, so my plan was to cover half of the poster just for the title. Yes, so I narrowed it down, and as the title of this session is Genetic and Database Resources Harnessing Diversity, I hope I said that correctly, I'm going to address both parts of my talk. So first of all, I'm going to present to you a data set that we've generated not only for our project, but also as a resource for the community. And in the second part, I'm going to give an example of how we utilize this to study diversity or the lack thereof. The project or the data set was generated in the frame of a project which had the objective of improving our understanding of not only the genetic control of a number of yield-related physiological, morphological traits in a population of European two-rod spring barleys. And in addition to just looking for, how genetic diversity affects traits, we also were interested in studying the role of gene expression networks. And the population that we used for this purpose was a selection of 209 cultivars and land races that were selected for the representativeness of the general European two-rod spring barley germplasm. I hope you can see that. This is each accession that we used is represented by a triangle on the right in the general space that is covered by the European two-rod spring barleys. This project was a collaboration between three partners: IPK Gattersleben, the group of Nate Stein, the James Hutton Institute, the group of Robbie Waugh, and the University of Minnesota with the lab of Gary Mulbauer. And the population was phenotyped in 2 years in the three partner locations with 2 to 3 reps per location. So we generated a wealth of phenotypic data, including 29 traits, which you can see on the left. And these traits include mostly earliness traits, such as days to on-tipping, days to heading, days to senescence, a number of plant height related traits, such as comb length, plant height, spike to comb ratio, many more. Number of grain size related traits, such as grain length, grain width, 1,000 kernel weight. Number of fertility related traits, such as the number of fertile florets per spikelet, and general morphological traits, such as growth habit. Now, Nils made a very good point of depositing data and making it available publicly. So this data is currently deposited at the Germinate database that Nils presented earlier this morning. Currently, it's available for registered users, but together with the papers coming out of this project, this will be made publicly available for everyone. We generated an expression data set coming from 6 different tissues, which you can see at the bottom. We sampled crown and root tissues from 1-week-old seedlings, developing inflorescences in peduncle tissues, and central spikelets right before flowering, and developing grains 5 days after anthesis. We performed RNA-seq on all the 209 accessions, so all accession by tissue combinations, and gene expression was quantified at the transcript level by pseudo-aligning the reads against the new Barley reference transcriptome version 2, which was just released last week, and which is a great new resource for gene expression studies in barley. And by utilizing transcript per million values, we're now not only able to make comparisons across individuals and tissues within our experiment, but also to link our data directly to other experiments and make comparisons there. The SNP dataset that we generated for this project was mainly coming from 3 sources, the most important one being the RNA-seq data that I just presented. And in addition, we did whole genome sequencing, whole genome shotgun sequencing, and the population was also phenotyped with the 50K SNP chip previously. So this provided us a wealth of data. Initially, we had about 40 million SNPs, and after filtering and imputation of missing data and LD-based pruning to actually narrow down the data set, we now have a SNP data set of 1.5 million SNPs, which I think is one of the largest SNP data sets that we have available in barley. The RNA-seq and whole genome shotgun raw reads have been submitted to the ENA archive and will also be publicly available later this year. And so will be the SNP data set. So this was just a very brief introduction into the wealth of data that we've generated, and it's not possible for me to go into all the details, but I'm happy to answer any questions you have about this during the breaks. But now I'm going to move on to give an example of how we utilize this data to learn more about our population and about the history of European two-row spring barleys in general. So we started out very simple with a PCA. With a PCA plot, and we can see that the population is kind of split in half, with a cluster of older accessions released mostly before the 1980s on the left side of the PCA, and younger accessions coming being released after the 1980s, approximately on the right side. This is illustrated here. And in order to identify genomic regions that were associated with this clustering, we ran an FST analysis along sliding windows along the genome. And we identified a region on chromosome 5H that is characterized by very high FST values, generally above 0.7, but you will see that in many cases it actually almost reaches 1. So that indicates that these two clusters carry complete different alleles, different haplotypes at this region. And this becomes even clearer if we look at the bottom, where we can see the frequency of the reference allele along the genome. So since all our reads were mapped against cultivar Barke, this is what we used as the reference allele here. And the newer cluster of accessions that were released after the 1980s is shown in blue. And you can see that on chromosome 5, it reaches a value of 1. So most of the newer accessions carry the Barke allele, whereas the older accessions, represented by the brown line, mostly carry a different allele. And this is also illustrated on the right, where you can see 10,000 randomly selected SNPs in this region of 250 MB, is the size of this region, plus 3 MB flanking regions on the left and on the right. And you can see that the cultivars at the top of this plot, these are the newer cultivars, and they all carry a red allele, and red means Barke, whereas the older cultivars at the bottom carry a different allele indicated in yellow. Now, there have been reports about high FST values between older and newer clusters of European barleys previously, but we now have a wealth of data available to further look into this and to characterize this region further. And one of the first data sets that we used was the expression data that we generated ourselves. And we wanted to know whether this clustering that we see is maybe also associated with differences in gene expression between the two clusters. And to do this, we used the gene expression data of the 800 something genes that are located in this region, and we performed an PCA on the gene expression. And we can see that the population is indeed clustered into mainly two main groups, with the older cultivars shown in orange on the left of the top left PCA, and the younger accessions carrying the other haplotype shown in green on the right side. And in the middle, there are 6 blue dots indicating 6 different cultivars that are forming a third, possibly even a fourth cluster. And to answer the question whether, or to identify genes that are responsible for this split into different groups, we used the same data, but this time on the top right, we plotted the genes instead of the cultivars. And we identified this group of red genes that you can see on the top left of the right PCA. This was a pattern or a cluster of genes that was always clustering separately from the rest of the genes. And this was a consistent pattern across all 6 tissues. So across all 6 tissues, these are 45 genes that have a specific gene expression. And you can see the gene expression in the heat map at the bottom. On the left part of the heat map, you can see the cultivars carrying the older haplotype, and these 45 genes are not expressed in this older haplotype, as indicated by blue colors. Whereas if you look to the right of the heat map, this is the cluster of cultivars carrying the newer haplotype, and here you can see that the genes are moderately or highly expressed, as is indicated by yellow and red colors. So we have a very clear on and off expression pattern based on the haplotype. And then we have the 6 cultivars that formed a separate cluster on the very right of the heat map. And here you can see that the genes do not follow this pattern. So they are indeed different, these cultivars. In addition to the 45 genes that are upregulated in the new haplotype, there was one gene that you can see on the top right PCA at the bottom. That's the only gene that showed the opposite expression pattern. So it was expressed in the old lines, but not in the new lines. I think that was very interesting. If we look at the development over time, we see that the oldest cultivar in our panel carrying the new haplotype is Union, which was released in 1955. And then if we look at the left plot, the green line indicates the increase of the haplotype in our population over time. So by the year 2000, it had completely replaced the old haplotype. I was going to say something about the right pedigree, but it's really difficult to see. I just wanted to point out there are the 6 cultivars that are that formed a separate cluster in the previous slide. 3 of them are related to Hordeum levigatum. So these 3 carry the Hordeum levigatum haplotype that was used in breeding in the 1950s because it conferred resistance to powdery mildew. And then there are 3 other cultivars forming a separate cluster, and they are not related to levigatum. So this is probably a different haplotype. Next, we utilized another database that we've already heard about a lot, and that's the IPK Gene Bank collection, which includes more than 20,000 barleys. And we wanted to look at the global distribution of the haplotype, of the new haplotype in this collection. And we can see very clearly that the haplotype is most frequent in accessions coming from the North African region. In the Moroccan collection, it reaches a frequency of 80%. And in addition to that, it's also found in some countries in South America and Central America. On the top right, we see just the land races from the IPK collection, and this is showing actually the same pattern, so it's most prominent in the North African region. And if we look at the bottom right, we can see the cultivars in the IPK collection, and we can see that there the haplotype is most dominant in Europe and in Mexico. And within Europe, there's a gradient, so it's not present at all really in the Northern European material, but it's most prominent in the southwestern Mediterranean region, where it reaches a frequency of about 50-60%. But these numbers are not entirely accurate because the IPK GeneBank collection doesn't include a lot of current cultivars. So that's why the percentage is actually lower than what you would have expected from looking at our population of two-row spring barleys. And this is actually, this haplotype is not only found in two-row and spring barleys, but also in winter and six-row barleys. So it's really widespread. So to summarize, I've given you a brief introduction into the multi-omics data set that we've generated. And this is now going to allow us to do a wide range of analysis, such as not only GWAS, but also transcriptome-wide association studies, EQTL analysis, and T-network analysis. These are currently ongoing and will be published later this year. And I gave an example of how we utilize this data to learn more about our European two-row spring barley population. And we've seen this haplotype on chromosome 5H that has risen so quickly in frequency in our population that it must most likely have been under selection for some reason. This region was not associated with any of the 29 traits that we looked at in our project, but of course we completely omitted the wide block of traits of disease resistance. So this locus, or this region, coincides with the RPH2 leaf rust resistance locus. So this is currently one of our working hypotheses, but we haven't actually tested this yet. However, I think this is a very important finding for breeding. We have this large region on chromosome 5 that is completely linked, more or less, and it has decreased drastically in genetic diversity in the European germplasm over the last just 60 years. So with that, I would like to thank a lot of people, which is my former group at IPK Gattersleben, my current group at the James Hutton Institute, and our partners at the University of Minnesota. And I would like to thank you for your attention. I'm happy to take any questions.
Thank you. So this area on chromosome 5 is very intriguing. Have you thought to do ATAC sequencing or something like that to see if there's a global suppression or activation epigenetically?
We have looked a bit into epigenetic regulation. We thought there was a clear pattern of methylation that would suppress expression in the old cultivars, but after having a second look, the picture is not that clear anymore. So we used methylation data from some of the pangenome lines. First of all, we had a set of just 3, representing both the old and the new haplotype. It looked perfect, but now I looked at a broader range of lines, and the picture is not that clear anymore. So.
Laura.
Hi, Ronja. I'm over here. So there's no recombination between the two, or do you think there's some kind of inversion, or is it just pericentromeric, and that's how it kind of keeps intact in this way?
We haven't seen any indication for recombination. And also these breakpoints seem to be quite conserved. There are a few lines, very clear from this plot, but there are a few lines that seem to have a different breakpoint in a way, so there must have been recombination historically. There was a second part of your question that I forgot.
Yeah, or is the recombination blocked because there is an inversion, or is because it's pericentromeric, so it keeps because there's low crossing over frequency, or?
There's no inversion there, surprisingly. This region is, there is structural variation right at the end, the beginning and the end of this region, if we compare the two haplotypes, but that so far is purely anecdotal. We have to look into more genomes representing the two groups, but might be a reason, that might be associated with the lack of recombination.
Thank you.
Thank you very much. Maybe it's time to come back, and I hope you are all refreshed to come together with me on this quest to find a candidate gene for barley development. So in this project, we are mainly focused on the shoot architecture, and the shoot architecture consists of many traits, for example, tiller number, leaf number, the number of nodes that are produced, but also the spike architecture. And there is this negative correlation between these vegetative traits, for example, tiller number, but also the reproductive traits, which is, for example, the number of seeds. And Aziz already touched this in his presentation before and said that there are, for example, some nutrient allocations that lead to this negative feedback, but also there are some genes that are involved in the production of vegetative traits and also in the production of reproductive traits, so at the same time having a dual role. Now the question is, can we use these genes to kind of break this negative correlations and try to improve yield with these? To do this, we first of all have to understand how the barley shoot architecture is set up. And for this, we need to know how the growth is regulated, because shoot architecture is a coordinated result of growth processes. And we also don't know really the key genetic regulators of shoot architecture development, and this is what I'm really interested in. And to do so, I'm making use of a forward genetics approach, and I use this really nice collection of barley mutants at Nordgen, and I'm mainly focusing on many noded dwarf mutants. These MND mutants have been already in part characterized, so we know the candidate genes for MND8, MND6, and MND1, and you can see they are all dwarf. They have massive production of vegetative growth, so they are high tillering. They also produce many leaves, as you can see in the seedling pictures, and they also have reduced seed set. These genes have been cloned with different techniques, for example, with marker-assisted cloning for MND8. This locus encodes a mate transporter family protein. The very first MND gene that has been characterized was found using reduced representation sequencing techniques, so exome sequencing, and it encodes the cytochrome P450 enzyme. And we recently found the gene underlying the MND1 locus, and I want to introduce you to these results briefly. So what we are doing in the lab is we are using RNA sequencing to look at possible mutations that can be found. And there are two things that are very important when it comes to this. First of all, you have to make sure that you are really selecting the right tissue and also the right developmental stage, so you can really see your gene being expressed in the RNA-seq. Now, when you have the reads, you can map them and compare wild-type and mutant sequences. And then in our case, we were working with the Bowman introgression lines. So we map the introgressions, we look for the candidate mutations, but we also look at gene expression. For example, if there's a gene that is heavily downregulated, this might imply that this gene is deleted in the mutant. Good. So we did this for MND1, and we found two major introgressions, one on chromosome 2 and one on chromosome 7. So these green lines are the alleles for MND1. And in the introgression on chromosome 7, we found the candidate mutation, which is an 8 base pair insertion in the second exon. And this insertion leads to a frame shift and a premature stop codon generating a null mutant. And this acyl-CoA and acyltransferase is not very well studied, but we are thinking that it is involved in the generation or in the modification of histones and thereby can result in different expression of downstream targets. So what does this gene do? We wanted to tackle this problem as well, and we looked very closely at the mutant phenotype. And here you see the main stems of the wild type and the mutant. And the triangles that you see are indicating the positions where the nodes are. And in the mutant, we have an increase in the number of nodes, but also we see something very interesting. For example, this white triangles that you see are representing the nodes where tillers have been rising, where tillers grow out. And this is in the wild type usually only at the base, at the elongated, unelongated internodes. However, in MND1, this was not the case. So we saw tillers arising throughout the stem, also at elongated internodes. And you can also see that the wild type was already flowering while this mutant line was not flowering. So it had a late flowering phenotype. Very close to the shoot apical meristem, we also saw something very interesting in the morphology of this mutant. So we saw that there were some axillary buds just below the shoot apical meristem. And looking closely using scanning electron microscopy, we saw that this is actually in the inflorescence. And we saw the formation of so-called leaf-like structures, which are usually bracts that are suppressed in the wild type, but in the mutant, they were growing out. And above these bracts, we have the triple spikelet meristem that later develops into the spikelets. But in this case, this triple spikelet meristem reversed back to a branch meristem, leading to the formation of tillers arising from the inflorescence. We also looked at the expression of the gene, and we found that it is expressed in young developing leaves. And now recently, we also found through molecular cartography data that the gene is expressed in the shoot apical meristem itself. So it really is expressed there where it is necessary to repress the formation of this vegetative growth. And this data is from my colleague Edgar de Mesa Arevalo. He has also a poster, and this is really great work, so please go and check this out. Now, we found MND1, we know MND6 and MND8, but can we maybe find more genes that are important for shoot architecture? So I was mining the MND collection at the Norton Data Bank, and we resequenced the 3 known genes, so to make sure we are not working on allelic lines. Most of the genes had mutations in the MND6 gene, so most of the mutants had mutations in the MND6 gene. But there were 5 that were still unknown. And of these 5, I want to present you one pair, which is MND-like 1 and MND-like 2. One of these is an X-ray induced mutant, the other is a spontaneous mutant. They are both recessive and allelic, and they are integressed into the Bowman background. Looking at the phenotype, we can see that this mutant is also dwarf. It only has 1 to 2 more nodes compared to the wild type. And it is also flowering just same as the wild type. So the phenotype of this mutant is not as severe as we can observe, for example, for the MND1 line. We started with our technique using RNA sequencing to identify the gene. And first of all, we were looking at the integration regions for this particular mutant. And in this case, since we are dealing with an allelic line, we really just want to see where's the integration overlap, because the candidate gene must be located in there. So this was actually on chromosome 7 at the same position where we observed the integration for MND1. So this was already quite interesting. But looking at the polymorphisms and expression data, we couldn't identify any candidate gene. So we went bigger and did whole genome sequencing. And we also tried to find the candidate here using different resources, also the PAN genome and reference-free approaches to identify the candidate mutations. But unfortunately, it was also not looking very good. So when these new techniques are not working, you go back to what is already quite established and what always works. And this is biparental mapping in an F2 population. So we had a very large population for MND-like 1. And at the top, you can see chromosome 7 with the 2 integrations as a scheme here shown. And we followed some CAPS markers in this population. What we saw is that there was a region in the overlap of these 2 integrations where there was no recombination at all. So that was quite interesting because this population was already very large. And we went back to our whole genome sequencing data, and there we found that there is indeed an inversion that is quite large of 3 million base pairs, including 132 genes. This inversion actually has a very interesting location. It is placed just in front of the known MND1 gene. So everything that is upstream of this inversion breakpoint was flipped to the other side. Then we also did fine mapping using the other allelic line, the MND-like 2 mutant, and we could not see such inversion indication of little recombination. But what we found rather is when looking at the whole genome sequencing data again at this region, that there's an insertion of roughly 300 base pairs. Now, having these mutations very much upstream of a gene is difficult because we don't really know what these regions do. They are not in a coding region and so forth. but we looked at the expression of the MND gene to find out what this probably cis regulatory mutations do. And we followed the expression of MND1 in young developmental meristems 5, 10, and 15 days after emergence. And you can see in the wild type, the expression is increasing, but in the mutant line, we don't see almost, or we see almost no expression for MND-like. So we crossed these two, and what we found, they cannot complement each other, meaning that they might be allelic. And the phenotype that we saw in the F1 plant was resembling the MND-like phenotype. So not the severe phenotype, but the rather mild phenotype. We further looked at the other MND mutants, and we found another one called MND7H, which also shows this branch phenotype. And we crossed it with the other MND mutants and found that also here no complementation was taking place. And if we have a combination of MND1 and MND7, so the new one, we see the phenotype of MND1 again. So what does this mean? We checked the expression of MND1 in this new line, and also here we found there is almost no expression of MND1, and the coding sequence looks pretty normal, so we cannot find any mutation. This might imply that this new mutant is also a mutant that carries cis variation in front of the MND1 gene. We quantified the phenotypes of the parental lines and also of the crossed F1 progeny. And I think this is a bit difficult to see, but I can explain it to you. So here we have the height of the lines. Gray is the wild type. These two green ones are MND1 and MND7. And this last one is the MND-like mutant. So these are the parents. And you can already see, depending on the allele that we have for this locus, the phenotype expression is quite different. And the different combinations between the phenotypes, so for example, MND1 and MND7H has a higher or makes higher plants compared to the other combinations when the MND-like allele is used. And we saw this gradual or this quantitative differences depending on the alleles for many more traits, not only for height, but also for leaf number and so forth. So this makes us conclude that, first of all, MND1 is a very important gene because it represses vegetative growth and ensures that the plant is developing normally. So it's really a key regulator of many different traits. And we found that there are cis regulatory mutations using the MND-like mutant and the MND7H mutant, and that these have really interesting effects on the expression of these phenotypes. So our hypothesis is that there's a dosage or maybe a spatio-temporal expression of MND1 that leads to this continuum of phenotypic variation that we can get in the shoot architecture. And with this, I would like to acknowledge Maria von Korff and also her group for this really nice project. And I want to mention Wilma van Esse, who actually started working together with me on this project quite some time ago, and our collaborators from Heinrich Heine University and University of Münster. And I'm happy to take questions.
Thanks, Agatha. That was very exciting, very nice presentation as well. So if I understood correctly, your MND1-like polymorphisms that you narrowed down are 120 kb upstream. So did you try to look into interaction of that region? Any loops, whatever? And I mean, in general, I think this is a very important study because it goes into the direction of what Zach Lippman has shown, that regulatory mutations in regulatory regions for extreme mutants may in fact be providing the variation we see in actual cultivars.
Yeah, thanks, Niels. I completely agree. So we are not so sure what this inversion really did. We I mean, it could have brought something more closer, like a repressor region, closer to the MND1 locus, but it could also lead to chromatin modification. So this region is going to be silenced around that. And what I didn't mention, but we have hints that this insertion that we see for the other allelic line, these 300 base pairs, are actually from a tandem repeat, which also often has an influence of the expression of surrounding genes. So we really think this could be epigenetic regulation then. So this is something definitely we would like to test next.
Yeah, it was related to that actually. So this 300 base pair insertion, is it also so upstream away from the gene, so it is not close by? So, I mean, you haven't checked if by any chance around that insertion there are any potential transcription factor binding sites, but it doesn't make sense because it is so far up away.
Yeah, it's really difficult because it's really that far upstream. And then if you try to identify transcription factor binding sites or any other motifs that are there, it's really a massive amount of things you will find. And then it's really difficult, I think, if you're dealing with promoter regions to really make sure that this has really an effect. Yeah, but the first thing that I would try to do is doing the chromatin analysis.
Do you have any indications from the protein function as to where the pleiotropic effect might be coming from?
So, from the protein function itself, I mean, maybe first about the protein function, we haven't proven that it really targets histones and thereby changes the expression of downstream genes, but there is evidence from rice that this is actually happening. This is something that we really would like to make sure. I think what is interesting is that MND1 is expressed in different tissues, for example, in the leaves that are initiated, but also in the shoot apical meristem. So I think it has a dual role, and in these different tissues, it might also have different downstream targets. So we would need to look a bit more closely there.
From the initial discussion that Niels raised, of course, with Lipin, you can, of course, do promoter bashing as a strategy. That's something that's easy to do. But I was curious about, from the high, if we're using Hi-C or ATAC-seq on this promoter, whether you will have a limitation at the end of the day for tissue, and how will you overcome that?
Yeah, this is also something that is a bit... it's a bit problematic. I mean, what definitely would be interesting to look at is using the MND-like mutant where we have the inversion, because the mutation is going to be in every tissue. So I hope to find the effect no matter which tissue I am looking at. If I want to see if MND1 itself is changing chromatin anywhere there, I would need to be very careful and look at the precise tissue where it's expressed. That's true.
Fantastic work, but a couple of questions. One is related to the actual pleiotropic effects, and the other one is on the trustworthiness that you can have for actual breeding of this. The one with the actual pleiotropic effect, do you think it is related, the fact that you reduce stem growth with the expression of exatelerine? And the one that's related to the application in breeding is, do you think that in a high stand density that crops normally are, the expression of the tillering capacity will still be seen there?
Okay, so two very good questions. So for the first one, this pleiotropic effect, I think it is, first I will separate it into vegetative and reproductive really. And the high tillering and the increased number of nodes, this all comes from the extended vegetative growth. So it has a really long period of vegetative growth and it initialized many, many leaves. So it's basically phytomeres that it's initializing. The high tillering comes then from the increased number of leaves because you have more potential axillary buds, but also it seems like there's there's some defect in auxin that tries to repress the outgrowth of these buds, as we can see in the wild type. For the use in breeding, so of course this plant, I mean, it's not usable at all, but there is actually a study in rice where they are looking at the homolog. And what they did is they overexpressed this gene, and they found really good traits. So it wasn't high tillering, it was low tillering, so exactly the opposite. And the grain size was increased. So I mean, this would be definitely interesting to see what will happen if we overexpress this gene.
Ladies and gentlemen, I wish you can see the slides and hear me clearly. First of all, I have to thank the IBJ's committee for offering me this opportunity to give an oral presentation. I'm glad to introduce you some genetic resources and a tilling platform. Phenotypic variation is the foundation of genetics. The lead scientist to functional genes. Then the functional genes provide breeders the most precise marker for selection. However, in natural population, a number of variants which are lethal or brings inferior phenotype in some special conditions would be rapidly lost. So, 100 years ago, scientists initiated studies on mutagenesis, intended to create larger diversity besides natural variations. During the past 30 years, loads of functional genes have been cloned from natural variants or induced mutants of barley, and many of them help scientists to do the genetic dissection of similar traits in other triticale crops. While the latest version of barley reference genome tells us that there are more than 35,000 of genes remains unknown. For barley, there are many mutagenesis populations have been developed, with most of their parent accessions had a European genetic background. For this study, at the beginning, we attempted to pick up a wild-type parent with East Asian descent. In Chinese, Ha-Tie-Xi is a barley landrace collected from northeast China by the National Crop Gene Bank in the middle of last century. It is with high recognition and is fully maturing. Except some lower part of the stems, the whole plant turns black, including all leaves, spikes, and all the nodes. Interestingly, when we look into its genetic background, we found it is distant away from East Asia. It may originate from Near East, but merged with something exotic. And we chose this genotype for another reason, that it is significantly susceptible to the barley yellow mosaic virus and the barley mild mosaic virus. We expect to isolate some host factors from the mutagenesis population. In addition, Ha-Tie-Xi shows outstanding grain quality with high content of protein and fiber, but low content of starch and fat, which is especially good for animal feeding. We first tested the responses of Ha-Tie-Xi to different dosage of the mutagene EMS. As the concentration rised to 40 millimole, the germination ratio of treated grains decreased mildly. but the fertility ratio of M1 plants decreased severely when the concentration rised to 30 millimole. So eventually we chose 2 levels of treatment dosage. We had 10,000 of grains treated with 22 millimole and another 16,000 of grams treated with 32 millimole. For the mutagenesis population, we obtained over 1,000 M2 DNA in stock and over 8,000 M3 seeds in stock. We investigated the plant growth vigor, fertility, and visible phenotypic alterations in the M1 and M2 generations. Among M2 lines, 12% of them that regenerated from 22 millimole EMS treatment, while 19% from 32 millimole EMS treatment had visible phenotypic alterations. Here are some examples of the phenotypic alteration. Maybe there are something you are just searching for. In order to better support the mutations discrimination, we constructed a Hatieshi genome assembly with multiple sequencing approach. Totally 3.8 gigabase sequences were assigned to the 7 chromosomes. Based on the Hatieshi mutagenesis population, we developed a TILING platform. A 3-dimensional DNA pooling strategy was applied so that increasing the screening efficiency. With the amplicon sequencing technology, multiple amplicons of target genes could also be pooled. Putative mutations were identified by calculating the normalized depth of the alternative genotype. Then, owning those mutations that simultaneously occurred in 3 pools at distinct dimensions each, the plate, the column, and the row were acceptable. For example, the SNP1 was detected simultaneously in the pools P8, C1, and R4. Through 2 independent experiments of this screen, that with 47 and 72 amplicons pooled in each, the mutation frequency was estimated as 1.5 mutations per megabase by 22 millimole EMS treatment, or 3.5 mutations per megabase by 32 millimole EMS treatment. Providing TILING service for many other research groups in China. To further increasing the screening efficiency, we used a 12 by 12 DNA pooling method. 3 subpanels covering 5,184 M2 individuals in total are included in the service. Through several batches of tests, we promised that averagely 21 mutants could be detected for every 1.5 kilobase fragment of your interested gene. Next, I want to give some examples to prove the value of this mutagenesis population in genetics research. The first example is a whole plant yellowing mutant. Because of its shortage of chlorophyll, the mutant grows much slower than wild type. However, it can get normal seed setting at the end. We crossed the mutant and the wild type to develop a segregated F2 population. Through backed segregant analysis, a genetic association was detected towards the centromere region of chromosome 1. In this region, a gene encoding for protochlorophyllide oxidoreductase was found having a prematured mutation in the mutant in comparing with its wild-type parent. And it is a little bit a shame that this one is not a new gene. It had been identified in another mutant before, in which a piece of deletion in the central part of the coding sequence resulted exactly the same phenotype as this mutant. We are lucky in another example. It's called schist spike mutant. The wild-type HTXi is a genotype with long uppermost internode. Its spike is above the ear of flag leaf, while the mutants had a shortened uppermost internode, and their spikes is below the ear of flag leaf. This phenotype is considered with a potential of increasing the number of spikes in a unit area. From couples of allelism tests, we found this pair of allelic mutants. We applied whole genome sequencing for these two mutants. It revealed 5 genes that had non-synonymous mutations are in common between these two mutants. On the other hand, backed segregant analysis was carried out within the F2 population that derived from cross between one of these mutants and the wild-type HTXi. An association peak was detected towards the end of chromosome 4, and within this interval, 3 genes were annotated with non-synonymous mutation in the mutant bark. The only one detected commonly through both approaches was taken as the candidate gene. We included this gene in a batch of Tilling screen. 7 additional non-synonymous mutations on this gene were detected. Some homozygous individuals in these mutation lines have been observed with schist spike phenotype. Functional studies on this candidate gene is ongoing. This mutagenesis population would also be valuable for reverse genetic research. As our experiments identified, about 40% of the mutations were non-synonymous, and prematured or altered splicing mutation were about 3%. Taking the barley semi-dwarf gene BR-insensitive1 as an example, novel mutations resulting decreased plant height have been obtained. In the future, we are interested in isolating more functional genes controlling developmentally or agronomically important traits. We are also picking up a subpopulation of mutants to form a corn collection and going to subject them for whole exome sequencing. Our research article is in publishing this month in the journal Plant Communications. This work is mainly carried out by the Bali Gene Resources Group that led by Dr. Yang from CAAS. Dr. Marshall from IPK and his colleagues provided bioinformatics support. We also get technical support from many other scientists from China and Germany. Thank you very much for your attention.